This lecture is going to work on Punnett square practice. A Punnett square is an easy way to look at a cross and figure out what the possible outcomes would be. If we look at this cross, we can see that we have a parent that has two dominant alleles up here and a parent with two heterozygous alleles here. On the left side, we could say this is the dad, and on top, we would say that could be the mom. So they each have two alleles, and because of the assortment, we are going to get different combinations. To do that, we would combine these two alleles in this box. So it would be big B, and then we would cross this allele and this allele and get big B, and then we move to the bottom. We move this big B and this little b, in this box, and we get the same thing here. So we would say that 50% or 1/2 of the offspring have a chance to be homozygous dominant, which homozygous would mean that they have the same allele, and heterozygous would be 50%, which means they have two different alleles. And when we're saying that it's homozygous, we want to make sure that we um, say that it's dominant or recessive because it's saying that it has two of the same alleles. Heterozygous, we know that the dominant allele is going to be the one that is shown in the phenotype. If we look at this a little closer then, we could talk about the genotype and phenotype. The phenotype is going to be the physical characteristics that you actually see. So if we said that the B stood for brown fur, so brown fur was dominant over, I don't know, we'll just say that little b is white fur. The physical characteristics here would describe just that, the color of the fur. The phenotype here would be 100% brown because of all the dominant alleles are going to be present in each of these possible outcomes. The genotype talks about the actual alleles that are present. So we would be talking about either the dominant or recessive allele. In this case, the genotype is going to be 50% homozygous brown or homozygous dominant, which are these two boxes up here, and 50% or 1/2 is going to be heterozygous because we have two different alleles present. If we do some more practice, and this is just a monohybrid cross, meaning that we're only crossing one trait. If we were to do a dihybrid cross, we would be crossing two traits, but we'll keep it simple. So we're going to use these alleles, the big T for tall and little t for short. One parent, we'll say, has a big T and a little t. They're heterozygous for the trait, but their phenotype would be tall. And the other parents will also be heterozygous, and their phenotype is tall. If we cross them, now practice this. So if you fill this box in, what are we going to get in this first box? You should have said big T, big T. The next box over here, big T, little t. And the bottom box, big T, little t. And hopefully you said little t, little t. So now let's look at the phenotype and genotype. The phenotype here is going to be 75% tall, because if you count 3/4, 3 out of 4, or 75%, express the dominant allele. And then 25% or 1/4 represents short, and they would appear short because they have the short allele. And we're talking about not people here. In this case, we would also still be talking about the pea plants. But we'll have short pea plants here because you have two recessive alleles. Now, what's the genotype going to be? Should have said that we would get 50% heterozygous because we have this box up here and this box down here that have two different alleles. Then 25% should be homozygous dominant or homozygous tall, and 25% should be homozygous recessive or short.